Hi, I am Maaly. I'm a machine learning scientist at ML ABI, and today I will talk about the machine learning framework I've developed in collaboration with Magnify and EuroPMC teams for discovering and enriching metagenomics metadata from open access research articles. Before diving into machine learning, I would like to give a brief background about metagenomics. What is metagenomics? Metagenomics involves the use of non-cultural methods to identify microbes in environmental samples. So basically, scientists go to different environments and collect biome samples from feces, water, air, dishwashers, clouds, insects, and then extract the DNA from these samples, followed by the sequencing and the assembly of these DNA, RNA contents. Scientists then use this sequenced assembled DNA to identify microbes in the sample. Hence the name non-culture methods, because they have avoided a lot of wet lab culturing techniques. Once the microbes are identified in the sample, they then investigate the link between these microbes and specific environmental phenomena or health or disease states. For instance, a couple of studies found a link between gut microbiota and diabetes or Alzheimer. Others found microbiota that can degrade plastics and fuel pollutants in marine water. Others found that microbiomes can produce antibacterial, antiviral, anti-tumor compounds which are beneficial for the drug industry. So to reach these conclusive metagenomic results, we need to uncover a lot of information about the environment, about the state of the environment, about the treatment given to the environment, about how the DNA was extracted, what kind of primers they have used. And this is really very challenging because microbiomes are everywhere. We are surrounded by microbes and we contain microbes. I usually use this slide to present the implications of microbiomes in a variety of environments. So this is basically the story of the green baby who refused to take a fecal transplant to become a marathon runner and decided to go to South Africa to get trained under extreme environmental conditions. And then he went through a series of microbiome unfortunate events. Then he returned back to UK. So the slide basically show that microbiomes in fecal transplants can improve health states and can treat diseases. Microbiomes can play a role in the biodegradation of plastics. Microbiomes can sterilize malaria-infected mosquitoes. And microbiomes can cause cancer. Microbiomes can treat cancer. Microbiomes can produce fuel in microbial fuel cells. And microbiomes can cause respiratory diseases like the black yeast in dishwashers and COVID-19. Microbiomes can be in clouds. Microbiomes can be in space stations. So how do metagenomic databases cover all this information? Metagenomic databases such as Magnify rely on the manual curation of microbiome sequences using metagenomic ontologies, such as the gold ontology or the environmental ontology. However, these ontologies cannot cover a wide variety of microbiome environments that I've shown you in the last slide, and their hierarchical level can lack a lot of important details for specific environments, which are essential for the metagenomic analysis. So the study on the left is a study retrieved from Magnify. It is about the effects of weaning methods on gut microbiota in horses. And as you see, the curators managed to tag this study with gold hierarchical level that has host-associated mammals, digestive system, large intestine, fecal assays. And as you can see here, even though all these tags are correct, they are kind of general because the study was in horses and there was no hierarchical level in gold ontology that fit into the study description. And also there are no tags for the treatment that have been used, like for instance, the weaning methods. Also, there is no information about what PCR primer they have used, what DNA extraction kit they have used. So accordingly, my current project Emerald aims for the development of an annotated named entity and automated named entity recognition that can extract the missing data about microbiome samples and environment from literature. So this automated named entity recognition, we aim to achieve using machine learning and deep learning. But whereas machine learning, deep learning pioneers offered unprecedented name entity recognition to train and implement, there were challenges. First, the database enrichment. How are we going to enrich the databases? Are we going to develop our own APIs? Are we going to to use current APIs or modify them? The second challenge was named entity recognition. So what kind of models are we going to use to train for recognizing entities? Which models are efficient? Are these models going to recognize the different microbiome environmental contexts or not? The third challenge was entity types and biocuration. What kind of entities are we interested in? Which entities are essential for the metagenomic analysis? And the fourth component was, or challenge was literature triage. So if we manage to pick up the most efficient name entity recognition model, and if we manage to successfully define all the entity types, what kind of literature are we going to use for training and curation? Are we going to select literature that cover a couple of environments, or are we going to select literature that cover a wide variety of environments so that we can train a robust named entity recognition model? So therefore, a machine learning framework was developed to enrich metagenomic studies with data about microbiome samples, environment, living organisms, and experimental methods from European C open access articles. This framework includes the following component: literature classification and triage, named entity recognition, and database enrichment. So we went for covering all the microbiome environment, and accordingly, I needed to pick up some classes that can cover all this wide variety of environments. So I picked up the top hierarchical level of the gold ontology, which has 3 classes: host-associated, environmental, and engineered. So to classify literature into these environments, I had to train a classifier. For this classifier, I had to construct a training data set. So how I'm going to construct the training data set? I know that Magnify already used gold ontology to annotate the metagenomic studies. So what I've done is I retrieved all the annotations from Magnify and linked them to the full text of their corresponding publications. This was my training data set. Then I used this training data set to train a multi-class random forest classifier to classify publications into host-associated, environmental, and engineered. As you can see, the metrics of the classifier were really good regarding precision, recall, and F-score. The next step was I would like to identify new metagenomics papers, not only the one linked to Magnify, because Magnify was kind of biased toward marine and human. So I wanted to cover more environments. So I've retrieved new metagenomic papers from linking ENA metagenomic studies to European C. I've retrieved around 14K ENA cross-reference metagenomic papers, categorized them into 3 categories, and selected a random subset of 140 papers that cover all the classes equally. And I manually validated this subset as a literature triage. The second component of the machine learning framework was a named entity recognition. But before recognizing entities, we need to know what entities are we going to extract or curate. So the first option was to go to ontologies, borrow some entities from there. But as I've mentioned, these ontologies don't cover a lot of environment, and they are so detailed regarding specific environment and so general regarding environment and hosts. So we came up with 16 novel metagenomic entities. These are ecoregion, engineered host, environment, sample material of the biome, the date of sample collection, the place of the environment or the host, the body site or the site of the microbiome, the state and the treatment of the environment or the host, and 6 experimental entities, including the DNA extraction kit, the target gene, the PCR primer, library strategy, library construction method, and sequencing platform. And I asked the curators, I defined these entities and asked the curators to annotate any term pertinent to these entities in the 140 papers using hypothesis. So after curation, I've done some post-processing on the annotations and tagged them with the annotation scheme bio. I've constructed 16 individual training datasets and trained each entity separately using BioBERT. So I've trained each entity separately because I have nested entities. Like for instance, body site can be nested inside sample material, like nasopharyngeal sample, nasopharyngeal body site, nasopharyngeal sample is a sample material. I've also performed grid search over 5 learning grades and 7 epochs to select the best model hyperparameters. We were interested in having more precision models than more recall models. So I selected the highest precision models for all the entities. As you can see from the entities, I have different learning rates for each entity. Each entity required different training epochs. And you can see also that the precision of all these entities were really good. And accordingly, I went for the last database enrichment component. As I mentioned before, database enrichment, in the database enrichment, we were wondering whether we are going to use novel APIs, current APIs, modify our APIs. Lucky me, I'm working with EuroBMC team. They have already annotation submission system and annotation API. So what I've done is that I've retrieved around 98K metagenomic publications using search query. And then I've annotated them using the name entity recognition model. I've submitted all the annotation using the annotation submission system, and all the annotations are now integrated in EuroBMC. You can see them using a highlight tool, which highlight all the annotation in the text on the front end, and also you can retrieve them programmatically using the annotation API. I've done also extra step, which is mapping all the annotations to ontologies to facilitate the categorization and publication using standard terms. And I've used a semantic ontology mapping tool developed by one of the teams at EMBL API called Zoomer. To enrich Magnify, now since we have provided all the annotation in EuroBMC annotation API, so Magnify can easily retrieve these annotations and include them as an enrichment layer in the databases. I've made sure to annotate all the publication linked to Magnify studies. Also, I've made sure to annotate all the publication linked to the metagenomic studies in ENA. And as you can see, the machine learning framework really did a great job in enriching the three databases with their metagenomic publications. I'd like to show you some examples of the metagenomic annotations. So here is a study about the ocular surface microbiome. And you can see here that the model identified the sample material, the body site as eye, the host individuals, the state, keratitis, and whether they were contact lens or not, the place, Miami, engineered, clinic, and DNA extraction kit, target genes, amplicon, and library construction method and sequencing. This is another study for the vaginal microbiome. You can see here the sample identified, and the body sites, cervical and vaginal and cervix, women, and the treatment of this woman was vaginal progesterone, also the kits, the genes, the library strategy, and the sequencing. And this is another study in horse where the feces are identified as a material, also the various body sites, the hosts, and so on. So another thing I was interested in is to integrate all the annotation in the Elixir Contextual Data Clearinghouse for cleaning and correcting all these annotations to be available for biosample and sequencing data resources. To our knowledge, this work represents the first large-scale automated enrichment for metagenomic studies. Machine learning and deep learning models were trained to enrich EuroBMC and magnify. And currently, I'm working on another machine learning framework to discover novel biosynthetic gene clusters in EuroBMC. I would like to thank Molecular Connection curators, San Diego from Magnify, and EuroBMC teams for their efforts in annotation, integration, and visualization. This work was funded by BBRC, and I would like also to thank you very much for listening to my presentation.